Thank you for the chance, and thank you for the Q-Nano conference to give me this opportunity to present my work. And my presentation titled as Human Primary Monocytes as reference cells for sizing the interference of nanoparticles with inflammatory defense reactions. Okay, we all know that our immune system can protect us from the dangerous signals, such as the fungal infections, viral infection, and bacterial infections. And the immune system has evolved to protect the integrity of body from damage, but its inadequacy will lead to pathology. So we do not know that if the nanoparticles will interfere with immune response. By far, we do not know that. And also, we do not know if nanoparticles will lead to the pathology even in a non-toxic doses. So that's why we should study this kind of issue. And if you want to study the immune system, you need to choose your target. We choose innate immunity because innate immunity is the first and immediate line of the defense, and also innate immunity very conserved and action system, and doesn't need to learn anything. It's always ready, and also the innate immunity doesn't undergo senescence of the response. This is the innate immunity. It consists by the epithelial barriers and phagocytes, and so also complements. And it will be react to the infections within hours, not like the adapt immunity within days. And within the innate immunity, the monocytes, or we call macrophage, is very professional phagocytic cells. And it can phagocyte the infections and produce aroused and inflammatory cytokines, and also release proteins to eliminate the infections. And also very important function is that the monocytes or the macrophage is a cell can do the function of the antigen presentation. And what about the response of the monocytes within the innate inflammatory process? I will show you in the following cartoon. There have tissue. If the tissue have infection, the resident macrophage will produce the CCL2. It's a chemokines. Then the chemokines will recruit the blood monocytes to active, to rolling, then penetrate the blood wall to go to the infected tissue, then actives as macrophage. Then the active macrophage will produce the chemokines, also very important pro-inflammatory cytokines, IL-1 beta, and some other chemokines, cytokines, TNF-alpha. Then the produced chemokines will recruit the T helper cells to produce very important inflammatory cytokines, interferon gamma. Then the interferon gamma will fully activate the mechanism of macrophage. Then the macrophage will be engulfed the infections to eliminate the infection. After bacterial destroyed, and the T helper cells will know that and stop producing the interferon gamma. Then the macrophage will be disactivated, produce some growth factors and anti-inflammatory cytokines. Very important one is IL-1RA to inhibit the inflammation and doing the tissue repairing. But what about the nanoparticles exist in all these processes? We do not know that exactly. So based on the previously described, we built a kinetic in vitro model for issue this kind of provide this kind of issue. We collect the fresh monocytes from the PBMCs, then culture the cells in different time. Then culture cells in vitro at different time. We give the different stimulation to mimic the real immune processes. From 0 to 2 hours, we give the CCL2 to mimic that the monocytes recruitment from the blood. Then from 2 hours to 14 hours, we will give the different stimulations. At first, we will only give LPS and TNF-alpha to mimic that early inflammation. Then till the 7 hours, we will add another stimulation, interferon gamma, to mimic that fully inflammation phases. And during this time point, we will put the monocytes into 39 degrees instead of 37 degrees to make that we have a fever in our body. Then after 14 hours, we will wash off all the stimulations and also use fresh medium to culture cells and also put the monocytes back to the 37 degrees to mimic that our body have a resolution way to eliminate the infection. And also we have other very similar model we call it persistent inflammation model to mimic that the pro-pathological ways. Only different is from 14 to 24 hours, we do not give the resolution way. Instead, we give the persistent stimulation. And within these two models, we use nanoparticles or solvent during 2 hours to 24 hours to mimic that nanoparticles meet macrophage in the tissue, not in the blood. And biomarkers we choose is L1 beta and its antagonist RNA. We use microarray to test different cytokines. And finally, we found that L1 beta and RNA is very obviously different at different inflammatory process. That's why we choose only these two biomarkers in this assay. Nanoparticles I use is, here I only show one nanoparticle, it's gold nanoparticles. Size is 10 nanometer and surface coated by sodium citrate. The concentration I use is non-toxic, 0.4 cm squared per ml and 1.28 microgram per ml. And first we saw the results. The gold nanoparticle in the first model, physiological inflammation model, which I give the resolution from 14 to 24 hours. And this is L1 beta gene expression only in the control group. We can see that during the recruitment from 0 to 2 hours, the L1 beta gene do not increasing, but from 2 hours to 4 hours, the L1 beta gene will be increased to the peak. And then from early inflammation to the late inflammation, 2 hours to 14 hours, the L1 beta gene expression will also be in a very high level and have slightly decreasing, but maybe it's not decreasing, it's only not significantly. And then go to the resolution rate from 14 to 24 hours. The L1 beta gene expression will goes down to a normal level. And what about gold nanoparticle and solvent? You can see here, it's very similar to the control goals, but have some inhibition during the full inflammation phases. But we also can see that the solvent also has the same inhibition. So maybe this kind of inhibition not due to the gold nanoparticles, instead due to the solvent. And the protein production. And we can see that from 0 to 14 hours, the recruitment and the inflammation phases, the L1 beta protein production will go increasing steadily and goes into the resolution rates. It will also goes down to a very normal level. And this is the gold nanoparticle group and solvent. We can see that it goes exactly the same as the control group. So we think the gold nanoparticles have no interference on the L1 beta gene expression and protein productions. And about its antagonist, RNA. Here I show the gene expression. It's a little different. From 0 to 14 hours, L1 beta RNA gene expression will go increasing steadily, then goes resolution rate because no L1 beta, so its antagonist will also goes down to the normal level. And here is the gold nanoparticle group and solvent. We also can see some inhibition, but maybe in the full inflammation phases. But as I said before, it's also due to the solvent, not due to the gold nanoparticles. And its protein production, the same as the gene expression, it will go steadily increasing from 0 to 14 hours, and then goes down to the normal level in the resolution rate. Also, the gold nanoparticle group and solvent. So we think the gold nanoparticles do not interfere with RNA gene expression and protein productions. And the second model, the pathological model, we give the persistent stimulation from 14 to 24 hours. We can see here, they have something different. From 0 to 14, it will go from 0 to 2 hours, no increasing, but 2 to 4 hours will go to the peak. And then in the inflammation phases, it will be not go down to the normal level, but will also in the very high level. And here we show some different. The gold nanoparticles in the 24 hours persistent stimulation will have some inhibition. They have some, but this is only the preliminary result, so this result we need to confirm. Also, the L1 beta protein production. We also can see some inhibition about protein production, but it's not a very dangerous thing. If you have some increasing of the gold nanoparticle group, you can go to the chronic disease. But if you have some inhibition, maybe not quite dangerous. So we think the gold nanoparticles may inhibit L1 beta gene and protein expression in the persistent inflammation. About its antagonist. And from 0 to 14 go increasing steadily of the R1R gene expression and also goes down to the normal level. I think because the R1 beta in a very high level, so the inhibits its antagonist production. About gold nanoparticle and solvent. Also show some inhibition during the full inflammation phases, but also due to solvent, not nanoparticles. And protein production. Here we can see from the 0 to 14 go to the peak, then 24 hours also in the same level. Because if you have a very high level of R1 beta, its antagonist also must very high in a very high level to protect your human beings. This is gold nanoparticle group and solvent. Also, from 14 to 24 hours will go increasing, instead of decreasing. So we think the gold nanoparticles do not interfere with R1R gene expression and protein production during the persistent inflammation. And we also do the R118 and N18 binding proteins. Here's the result. You can see that the gold nanoparticles and solvent also do not have inhibition or any interference in both the pathological inflammation and persistent inflammation. We also do others. Because of time, I cannot show everything. We also do the soluble R1R2 and R6, R6. These two factors do not show any effect. But R8 is seen to be a partial inhibition during the full inflammation phases only. But also we need to confirm this result. So I will give the conclusion now. And we built a kinetic model describing the monocyte reactions during the course of inflammatory response, which can be used to assess possible interference of nanoparticles with the innate immune reactions. And the model is in vitro, and we can replace animal use. And also some results from animals, not that from the human beings. So the best thing to use the human cells. Also, this model is based on the human blood monocytes. So the result we got should be highly reliable and can represent the human response. Specifically to our gold nanoparticles, the gold nanoparticles do not significantly interfere with either the physiological or the persistent inflammatory response in this in vitro model-based kinetic assay. And acknowledgement. I want to thank my supervisor, Diana Velasky, and my two colleagues, Paula Italiani and Paula Giungato in Pisa, Siena. Also our collaborations, Professor Puntas from ICM Barcelona and the UDOT Noctran. And I also want to thank Professor Dorsch from Salzburg University and Professor Miglionini in Pisa University. And this study is supported by the European Commission and Marine Corrections and Nanotools project. And thank you for your attention.
Thank you for this very clear and nice presentation, which is now open for questions. Yes, please. Microphone.
Hi, my name is Tom Sterk, coordinator of NanoReg. Interesting. I was really wondering what type of control were you using. I'm asking this because you look at the inflammatory response, but you probably know that in the old days, rheumatism was treated by colloidal gold in order to suppress the inflammation. So you might, from a nano perspective, it's nice to use gold because it's so persistent and it's easy to detect. But there might be a beneficial effect of the nanoparticle. And have you controlled that out?
In our model, and for this model, I just use the fresh monocytes as control. But specific to the gold nanoparticles, we just use the gold nanoparticle as control and the solvent as another control. We do not use other controls.
But it is an interesting point. I mean, when you discuss the fact that in the persistent model, the gold nanoparticles, you mentioned that the gold nanoparticles may interfere. But I think the point is that maybe the gold nanoparticles can be considered to have a beneficial therapeutic effect, much like gold salts in the old days.
Oh, yeah.
So gold salts could be a
result. I think this result is only a preliminary result, and we cannot confirm that this inhibition is only from the gold nanoparticles. We also need to confirm this result. So I just show here is only a preliminary result. And also, and I will take your comments and to use another control to show that. But from my previous experiences, we do not think that GO nanoparticles will be really inhibit inflammation during the full inflammation phases. We think GO nanoparticle is non-toxic.
There may not be enough time to develop into a long-standing, persistent inflammation which would have been treated with gold. So the 24-hour window.
Yeah, and also this is not the full model; it's an easy model. And we also know that T helper 2 cells will produce some other cytokines, IL-4, IL-13, to inhibit inflammation. But here I do not show that because I do not use in our model. So it's only an easy model to modify that the inflammation during the phases in the human beings.
I agree that it's very important to use human cells. I mean, there was a publication very recently in PNAS with a very large-scale transcriptional profiling showing that mouse models completely failed to mimic the human inflammatory condition. So it is something to bear in mind, and I think, therefore, I appreciate your model.
Yeah.
So if no further questions, let's move to the next, I think, final speaker before lunch.